In this video, we're going to think about how a nerve impulse passes from one neuron to the next. The electrical impulse needs to pass across the gap between one neuron and the next. Notice here that they're not actually touching, and it is that junction between one neuron and the next that we call a synapse. I've circled it here. You can also find a synapse between a neuron and either a receptor cell or an effector cell. An example of a receptor cell might be, say, a skin cell that receives information, for example, about temperature or pressure. An example of an effector cell might be, say, a muscle cell, where there would be an effect produced, such as movement. So how does the nerve impulse or action potential pass across the synapse? Let's look at the synapse. So first of all, we have the pre-synaptic neuron, and that's where the action potential is coming from. And then we have the post-synaptic neuron, and that's where the action potential is going to. And both of these are enclosed by a membrane, and that's the yellow line around them. The presynaptic neuron has these vesicles that are filled with a neurotransmitter, a chemical. And then we also have calcium ions present outside of the presynaptic membrane. So when an action potential arrives at the presynaptic neuron, it means that the presynaptic neuron has been depolarized, and the action potential is represented here by the red arrows. A sequence of events then begins, and the first of these is that calcium ions diffuse into the terminal buttons. This causes the vesicles containing the neurotransmitter, for example one is called acetylcholine, to move towards the plasma membrane of the presynaptic neuron and fuse with it. They therefore release the neurotransmitter into the gap between the presynaptic neuron and the postsynaptic neuron. We call this the synaptic cleft. The neurotransmitter diffuses across the synaptic cleft. Once the neurotransmitter reaches the postsynaptic neuron, it binds with receptor proteins shown here in pink that can be found in the membrane of the postsynaptic neuron. The binding of the neurotransmitter to the receptors in the postsynaptic neuron causes sodium ion channels in the postsynaptic membrane to open, and sodium ions then diffuse into the postsynaptic neuron. If enough sodium ions diffuse into the postsynaptic neuron, a threshold potential can be reached. The postsynaptic neuron is depolarized, and therefore a nerve impulse can be initiated along it. The neurotransmitter is then degraded or broken down by specific enzymes and released from the receptor protein. The sodium ion channels then close, so no further sodium ions can diffuse into the postsynaptic neuron. The degraded neurotransmitter fragments can then diffuse back across the synaptic cleft, and they are reassembled in the terminal buttons of the presynaptic neuron. In this way, the neurotransmitter can be reused the next time a nerve impulse or action potential needs to be transmitted across a synapse.